today, so the slide's not up anymore because it goes to my computer, but today we're focusing on the flow and distribution networks. So yesterday we talked about complex spatial networks in a sort of general structural setting. What kind of metrics can we use? What kind of metrics from traditional network science aren't so useful? What are some examples of metrics that are one purpose builds for complex spatial networks? And so now, that's sort of, yesterday was sort of mostly taking a step back and was thinking purely about the geometric topological structure. And now today, we're going to focus a little bit more on, networks, complex spatial networks with a more specific function, right? And then kind of also think about how to incorporate that function into the way we're thinking about things. And so, because this is a really broad audience, so it's going to be flow and distribution networks. So think, vasculature in the body, or river networks, or the leafination, channels that I showed in the early slides from yesterday. And I think there's people from backgrounds from like non-physical science backgrounds as well in complex system studies, economics and things like that. So I think it's worth talking a little bit about fluid dynamics. So that's where we're going to start. Blackboard for a little while, then I'll jump to slides. We'll have a break, and we'll have a couple more topics on Blackboard today. So, we're going to start with high nuclear cell flow. How many people know what that is? A couple of people, but not many. But okay, so that's good. Perfect. Okay, so high nuclear cell flow is sort of like I'm sure you've heard of the Navier-Stokes equation. The Navier-Stokes equation is sort of like the general, nasty, horrible, highly nonlinear equation that describes sort of all of fluid mechanics. It's notoriously difficult to solve. There's a millennium math problem on, proving things about its stability, things like this. But for many sort of less general, more specific practical scenarios, there are different either linearized or simplified models of flow that are much easier to work with than full-blown Navier-Stokes. Okay, and so how can we say is one of these? Right, it's the idea is to describe flow in a pipe or a channel. So you have some flow going in, and maybe it has dynamic viscosity, we'll call it mu, right? And you have some channel, which we're going to assume to be a cylindrical channel, like a pipe, literally like a pipe. We'll call radius capital R, length capital L. Out. The pressure, the inlet we call PI, the pressure in the outlet of the pipe F. And then of course we can write for convenience delta PI. Right, so just the pressure drop, over the course of the pipe. And so then we want to work for the derivation of how we're going to get to this simplified setting. We're going to work in cylindrical coordinates, and we're going to call this coordinate r along the pipe x, and then the azimuthal coordinate we're going to call theta. Okay. So the question then is, what is high nuclear cell flow describing? It's describing, flow in a situation like this that is laminar, right? So we're also going to assume laminarity flow. So that means not turbulent, right? So turbulent flow, of course, is much more complicated. So we're going to stay away from that for now. We'll sort of phenomenologically we include some properties of turbulent flow a little later, but for now we're going to just stick to laminar flow. Okay, so the starting point is Navier-Stokes. Right, so in particular, the Navier-Stokes momentum equation in cylindrical coordinates, 3D cylindrical coordinates. And I'm not gonna write those down because these two boards are probably not big enough to write them all down. They're terrible, especially expressed explicitly in the coordinates. It's not great. But fortunately, we're gonna make another, despite the fact that we are having this specific setup, we're gonna make some more assumptions. Right, and that's gonna help us, actually write down much more sensible equations that come from that stuff. So we're gonna assume the following things. We're gonna assume that we have, maybe I should have a different color for each. We're gonna assume that we have steady flow. Right, so what does that mean? Right, that means that all the possible time derivatives of the different quantities in the system are zero. Right, so we're at steady state. Right, we're not thinking about any transients. We're not thinking about like the buildup of flow. It's not that we like suddenly turn on a faucet and are like looking at the transient buildup of the flow. We're thinking about, once we're at steady state, once we have steady flow, time derivatives are all zero. Of course, that kills a lot of terms, right, in this set of equations. So that's good. We're also going to assume that we have no radial or azimuthal flow in the pipe, right? That is to say, the flow direction, if any location in the pipe, you're flowing down the pipe, but you have no component of flow around, right? You don't have a vortex in the pipe or something like that. That would be a nightmare. You're also not flowing, any little sort of element of fluid in the pipe is also not flowing, radially, not in or out. Like it has some profile, perhaps, speeds, but they're all pointed forward. They're all going down the pipe. Okay. So again, for most reasonable pipe geometries, this is actually a pretty good assumption. In particular, what this means, right, is the radial and azimuthal components of the flow are zero. Okay. And then again, we'll kill some more terms, right, from the general Navier-Stokes set of equations. That's good. The more things that are zero, the more terms are going to go away. Okay, next assumption is the flow is axisymmetric. Right, so that means that any derivative of any quantity in the system with respect to the azimuthal coordinate would also be zero, right? So that everything has, axisymmetric symmetry, right? The pipe has axisymmetric symmetry. We're gonna assume that there's no, we're also in a laminar, we want to treat a laminar regime. So we're gonna assume that there's no spontaneous symmetry breaking, there's no funny business going on, right? Things are in the sort of simple linearized regime. And so again, that regime, this turns out to be a very good assumption as well. And again, because now we're setting a whole bunch more derivatives to zero, kills more terms up here, right? Again, literally, if I write this out, it would probably take out both words. So we have lots of terms to kill. And then one last assumption. We're also going to assume that we have fully developed flow, which sounds like steady flow, but it actually means something slightly different, right? It means that the flow profile in the x direction along x is also constant, right? So that you don't have, this would be something that may occur as you're building up, but really this is equivalent to some of the previous assumptions together with mass conservation, which of course is not an assumption, it's a requirement. Right, you don't want to have, you have spatial gradients in the flow, then you know, somehow are having fluid appear or disappear somehow. Which, you know, if it's lossless, if it's not a leaky pipe, right, and you don't have, you know, you don't have little inlets or something that we're, you know, suppressing or something like that, then this is also a function. Okay. And so now with these assumptions, it turns out that the negative spectral momentum equations, right? So there's one momentum equation for the angular momentum, for the radial momentum, and for the axial momentum, right? So for each of the coordinates, right? And so then the angular momentum equation just reduces to identity, right? So it loses, you know, it loses any meaning, right? So you don't have to, it drops out essentially, right? Like you write down the angular momentum equation and you make the substitutions that you get from this, and you find, you know, 2 equals 2, right? It's not that simple, but you find something equals itself, right? Essentially. And so, okay, great. So that one we don't have to worry about anymore. For the radial momentum equation, everything reduces down to the radial profile of the pressure equals 0. Right? So there's no pressure change radially in the pipe. Okay. Right. So now we also know already that there's no azimuthal change, you know, the pressure in the pipe either, because we already assumed that everything's axisymmetric. Right. So that means that the only way the pressure could be changing is axially. Right. And then, so that leaves us with the axial momentum equation. The only one that's interesting at this point. I mean, this has information in it, but it's not, you know, so interesting. Right, so what's this? This ends up being, oh, I'm trying to take this out. This ends up being, right, the following thing. So you get something interesting from the axial equation. So you have, you know, some function of r's and derivatives of r's applied to some operator essentially applied to the flow in the x direction, the differential operator there, and then that's equal to 1 over the dynamic viscosity times the change in pressure and as you go down the axial direction of the tube. Okay. And so what can we do with this? Well, we can be clever. We can observe the following facts, right? So this whole side of the equation is a function only of r, right? So how do we know that? Well, there's only r's in the differential operator, right? And we know that ux, right, the flow in the x direction is not dependent on time, because nothing is dependent on time, and not dependent on the angle, because nothing is dependent on the angle. And what else might it be dependent on? It could be dependent on r, right? Or x. Those are the other things left. Oh, wait, it's also not dependent on x, right? So it's only dependent on r, right? So ux is only a function of r, right? And the differential operator is only a function of r, right? So this whole left-hand side of the equation is just a function of r, right? Now, meanwhile, the right-hand side of the equation is only a function of x, right? So now the operator, which is one derivative, is a derivative of x. This is a constant. And now the pressure, right? The pressure is not a function of time because nothing's a function of time. It's not a function of the angle because nothing's a function of the angle. And from the radial momentum equation, we know that the pressure is not a function of the radius, right? So x is the only thing left. So pressure is a function of x, the operator is a function of x, the whole thing is just a function of x, right? So the left-hand side and the right-hand side are functions of two completely separate, distinct variables, right? It means each side is a constant, right? So how can a function of r be equal to a function of x? Well, if they're both constants, it's the only way that can be true, right? So now we know that each side of this is a constant. And so then we can figure out what that constant is from the right-hand side, right? We can write that minus, just for a nice easy sign convention, minus the PDX equals delta P over L, right? Because now this is the change in pressure in x, right? And now we know from the setup that we assume we have some pressure in, some pressure out, and some change in pressure over the whole course of the tube. And so we're going to just take the linear approximation here. Like in principle, that pressure profile, it doesn't need to change, linearly down the tube. But this, assuming that this is the case, turns out also to be in the laminar flow case, turns out to be a good solution. Okay, so the profile of pressure down, and actually if it weren't like this, you would still have some x dependence left over, and you wouldn't be a constant. So it actually has to be linear. It's not even an assumption. Forget I said that. It has to be that way.
You can just integrate it.
Yeah,
The
exactly.
boundary condition.
It's a boundary condition, that's right. You can just integrate it.
Then you get the result.
You get the same.
So it's nothing.
No tricks.
There are
No, this is
no assumptions in it.
Yeah, exactly right. And then we can solve for u. Again, just by integrating. So u of x now, and then taking advantage of the fact we know what the constant is going to be delta p r squared over 4L mu plus C1 log R plus C2. Okay, so you carry out the integral, you get the following expression with two constants of integration that you don't necessarily know what they are. However, right, remember R is the radial coordinate. So this term blows up at R equals 0. And we don't want the flow to go to infinity at the middle of the pipe. That seems unphysical, right? So then that means that this C1 has to be 0. And this whole term goes away. Okay, and then what else do we know about the flow? Well, we want to assume no slit boundary conditions, which again is, a quite typical reasonable assumption for fluid flow in a pipe. What it means is that the flow velocity on the boundary of the pipe has to go to 0. Right, so that means that for little r equals big R, ux has to be 0, right? And so the no slip boundary conditions means that C2 has to equal delta P big R squared over 4 mu. Right, and in the end, okay, flip the sign somehow. In the end, you get a nice parabolic velocity profile in the pipe, which is generically true for flows in pipes, linear flows in pipes. Ux equals P over 4L mu times R squared minus R squared. Okay. Stops that. Now, almost done, but it's not going to fit on one board quite simply. But to finish, we observe that the maximum velocity in that profile, of course, is going to occur in the center, right? So R equals 0, so U max is equal to U of X at R equals 0. And it is equal to delta pi of G capital R squared. Yep. Okay. And the average velocity at any position along the pipe is equal to cross-sectional area normalized divided by integrating around the rings of what the value of U is around the rings. You perform this integral, and it gives you the average velocity is just 1/2 the maximum velocity. Okay. Okay, and now the last step to get where we want to go for the Hagen-Bussell equation is to say, all right, well, you're actually making a measurement, right, of what's going on in a pipe. It's very difficult to like measure the flow rate at a specific position inside the pipe, right? What is typically done is that you see how much volumetric flow is coming out of the pipe at whatever rate, like how much volume fluid comes out at a certain rate. Right, so let's write the volumetric flow rate. This is the more experimentally accessible, meaningful quantity. Q. And so what's the volumetric flow rate? Well, it's got to be the cross-sectional area times the average velocity, right? So it's pi r squared times the average flow velocity. Right, so that's going to be the volumetric flow rate. All right. And what is it? Well, you can just substitute everything in that we've gathered up now. And I can write it for Q, but actually traditionally you then flip it around and write it for the pressure drop. We'll just go straight there. Okay. That is the Hagen-Bussell equation, right? It linearly relates the pressure drop in a tube or an edge in a network to the volumetric flow rate, and this sort of combination of factors to do with the geometry and the size of the pipe and the flow and the viscosity, right? And so this is a pressure drop, right? This is a flow, right? This actually might remind you of the following equation, where V is not volume, but voltage. I'm pretty sure this equation is on this funny board somewhere.
Yeah, some resistors.
Yeah, but this is equivalent. This is formally equivalent to Kirchhoff's laws at this point, right? Where you now replace the resistance in the Kirchhoff circuit with this sort of geometric combination of terms, right? That interestingly goes like has this r to the fourth dependence, right? So if you make the tube bigger, right, then the resistance goes way down really fast, right? Like it's much stronger to make the tube, the cross-sectional area bigger than it is to lengthen. Like lengthening the tube makes the resistance go up, but just linearly. Whereas, widening the tube really makes it dive the square of the area. And all the other Kirchhoff relations, and you still have conservation of mass in the flow is the same thing as the conservation of the electron flows and everything. All the Kirchhoff's laws actually go through the same way, because of the fact that you have this formal mathematical identity between the gradient of your pressure field and the flow, right? And you have some linear term sitting between them. So for example, this is going to come up a little later, right? Like the power consumption in a tube like that. And then, yeah, so for example, capital P, not pressure, capital power consumption is UL over pi fourth squared, which again is like power equals R I squared. Or I squared R. Usually it's I squared R for whatever reason. It's written. I'm quite sure that this is on the thing somewhere. Yeah, so this one is going to come up again later. And the fact that this is just formally equivalent to the Kirchhoff circuits is really nice, right? Because the Kirchhoff circuits, they're linear, they're relatively easy to sort of work with. You have, well-defined conservation equations. So if you then blow this up to not just thinking about a single tube, but thinking about a whole network of tubes, right? You can still have, you still have the pressure drop right along the tube. You still figure out how much flow goes in, right? You can basically, for any given state of your conductances, or your resistances, conductance being one of the resistance, right? You can determine the global flow profile from the topology of the network. And the sort of spatial embedding, of course, the spatial embedding comes in and right in the L's and the R's and the fact we have all these things. And so now that we have this sort of setup, we can think about adaptation of networks. Right? If you have some flow network and you have some feedbacks, right, that depend on the state of flow in the network, right? Let's say, you have some biological system, and I'm going to move to slides in a second to show some examples, but it's not so crazy if you imagine some biological system that has a huge amount of flow, right? Like a very high amount of flow, high pressures in the system, right? It might want to make R bigger, right? To reduce the power consumption, right? The power consumption goes like the flow squared, right? But also goes like 1 over R to the fourth. So you have a lot of flow, that means you're burning a lot of power. But if you make the conductance, more conducive to allowing the flow, then the power consumption is going to go down, right? And so let's talk about what we can do with that. So I'm going to switch to slides until the break.
Just switch the camera on.
Yeah, so I guess I need to switch the camera somehow to the. I need to sign back into. I'm going to sign the Zoom.
Oh, because now it's only the Zoom. Oh, switch to the.
Yeah.
Yes.
Can I ask a question in the meantime?
Of course.
This Kirchhoff law kind of thing, isn't that more general than only for Hagen-Poiseuille flow? So I can't imagine it not being true, because the pressure along a loop, if you get back at the same point, the pressure is the same, so the change in pressure should be 0, and there's mass conservation at.
Yeah, so those parts of the set of Kirchhoff laws those are more general.
Yeah.
But the specific relationship between the flow and the pressure drop, for example, if you have turbulent flow,
Yeah.
right, then the scalings become very different.
Yeah, so it's not
So the conservation rules, both of like the fact that the pressure or the voltage come from some conservative field, the gradients in a conservative field, and the fact that you have conservation mass, those are quite general.
Yeah.
But this linear relationship between the flow and the pressure drop, the voltage and the current, that depends on the set of assumptions that we work through that are, reasonable in many cases, but they're assumptions and they restrict you to certain cases. Okay. It's very consistent. No. Right, and then.
Share screen.
Yeah. This. This.
For this, the recording is nothing.
yeah, I just don't want to block anything on that because it's on here.
Alright.
Okay, anyway, so yeah, so now to the slide part. Flow distribution networks and talking hands. Okay, so just to remind you, that we had the slide already yesterday, just as a quick reminder, right? Like, I'm interested in, and we're going to talk about these 3 days, is the cases where geometry and topology both are found together, right? So we're going to be interested in now thinking about taking this setting and blowing it up to, to the complex spatial networks, but now we're including geometry in the sense that the network is embedded in space, right? And so it has things like real lengths and real radii, and it still has some interesting topology, potentially. And this was all same slide. In the back of your minds, we want to be headed towards, biological structures like this. Okay, but now let's talk about adaptation, right? So the idea is that if you calculate, you have some flow in on a node, in this case, so ignore the hexagon on the bottom for the moment. So in the hexagon on the top, we're going to inject flow into the middle, and then we're going to have some sinks sort of on every other node, some small pressure drop sinks sort of everywhere, and a large pressure drop source in the middle. And then we're going to say that we can calculate the wall shear stress, right, that a certain amount of flow gives, from the Hagen-Poiseuille profile, right? So essentially you have the slope, right? The flow is 0 at the walls, but the slope is not 0. And so that induces a stress on the walls. And that stress, produces, it produces a signal essentially to the system that, you want to reduce the power dissipation in the system, you can reduce the wall shear stress. Like it's related, that stress is also related to the power consumption in the system. Okay, and so this is the update rule, right? So we're going to change the radii, in a way that minimizes the wall shear stress, but there's this offset, right, that says that there's some net cost, right, of upkeep. So that like if we only had minimization of the wall shear stress, we would just make all the tubes as big as possible, right? Every single link in the network, we would just have R going to infinity, and we would certainly minimize the wall shear stress. But if you include then a cost of having a larger and larger tube, right, then this becomes a much more interesting adaptation rule. So this kind of rule was first written down for adapting network structures in slime molds. So some of you may be familiar with this Polycephalum physarum slime mold. I think I have a movie of it, but I don't think I have it in the talk. Maybe I can find it for the afternoon session just to show it. It's really cute. Where the things can solve mazes. There's very highly adaptive networks that the slime molds make, where they go foraging and then they can sort of reduce onto a complex network, and they can reproduce the subway system in Tokyo, given the right, boundary conditions and the crazy cool stuff. But anyway, so this kind of adaptation rule is first written down for that, but it's found to be somewhat more generally applicable to other biological adapting networks. And so, okay, so what happens with this adaptation rule in the system? So we'll let it run. So we start with very big flow everywhere, and big, the thickness is the conductivity. Okay, and so so it's sort of, again, so I said we're injecting flow in the middle, and we have outlets everywhere else. And so the adaptation worked, right? So you start, making, some edges get smaller and smaller and smaller, and some edges stabilize. And interestingly, in this case anyway, you ended up with a tree, right? So we're interested in complex spatial networks, and I told you yesterday that complex could mean anything, but to me it means that there are cycles. And so there are no cycles, right? And now this is not an accident. One can show, and actually classically, even I think in the 19th century, people showed that if you want to optimize transport sort of generally from one location to multiple locations, or vice versa, then, and you have some cost of the pathways over which you are carrying the transport, then a tree network is always the best way to do it, right? And so here we have one inlet and multiple outlets, and we got a tree, so that's good, I guess. But think about the leaf example I showed yesterday, or these 3D complicated vasculature in organs, there you typically have one inlet and the capillary bed sort of goes down to many, many outlets. And yet those systems are highly ramified, right? You have many, many cycles, you have much more topological structure. How could that be? Like, how could you normalize, how could you stabilize the loops, okay? Because this is actually generic. Like we could run this many, many, many times with many realizations of the sources and sinks, with many values of the strengths of the sources and sinks, always be true, no matter what. So the answer is noise, is fluctuations, right? Or at least an answer is noise. So it turns out, we're covering the paper, unfortunately, the rest of the course is the first author in the year 2013, which is enough, but unfortunately, the movie of the upper one is covering the rest of the citation. Anyway, the idea is that you do the same thing, but now instead of calculating a specific set of pressure drops on every link, you now calculate, you do many, many realizations with fluctuating sink and source strengths or locations, right? So you calculate, what is the flow profile for this set of sources and sinks? And then I do it again, I do it again, I do it again, I do it again, where each time I draw a new set of source and sink weights from some predetermined distribution, Gaussian distribution, or some other distribution, doesn't actually matter because of the central limit theorem, if you do it enough times. And then the update rule only sees the average of the pressure drop, the average of the power consumption over all those realizations. So the idea, what is the idea here? What is the underlying? This seems a little weird, but there's a very clear physical principle here. What this means is, all we're saying is the adaptation timescale in the system is slower and slower in a way that's timescale separated from the timescale over which you have natural fluctuations in the flow patterns, right? And those fluctuations could come from many different sources, right? They could come from changing demand in the network, in a biological network. They could come from changing, actual supply on the inlets, right? It could come from, natural fluctuations in the upkeep processes or the cellular processes that live on the whatever tissue is around the system, right? And many different reasons why you might have fluctuating, conditions in the system. But what this is saying is that the update timescale is slower than those, right? And then it makes sense, physical sense, that you would have something like this. And if that's the case, if the sort of magnitude of the fluctuations is high enough, that's the standard deviation versus squared versus mean squared, right? Then again, you simplify the network quite a bit, but you stabilize a lot of cycles in the end. And it actually becomes quite interesting, how many cycles that you could have had versus how many you do have, depending on the strength of the noise. If this noise goes below 1, then you still go back to a tree even with noise. So it has the properties of a second order phase transition, where the number of remaining cycles is 0, 0, 0, 0, and then you have non-zero derivative and it jumps up. So there's a lot of interesting structure to this. Okay, but still, yeah, questions?
So in the noise term, do you also change the position of the source?
Good question. So one could do that. So there's the specific implementation that Corson used and also that we used later for some of the stuff that we did was we kept the fact that there was one source in the middle and everybody else was a sink. And we changed the magnitude of the sink, pressure drops randomly. Okay, but the same thing happens, and it's been shown by other people, that the same thing happens if you just randomly move around where the sources and the sinks are, or if you do both at the same time. Like, just as long as the time scale of whatever underlying fluctuation it is, if it's the magnitudes, if it's the locations, so long as you just do lots of realizations, right, and update slower than that, then it still works. But it's a good question, and I think likely some of the details of like the phase transition and the slope and where it occurs can be shifted a little bit by the specific, like, is it like location noise, is it magnitude noise, is it a mix, like those kinds of questions. Yeah. Okay. Think about, right, like another way to think about this noise thing is think about traffic in a city, right? Like, if everybody always were gonna go from from their home to, an office building downtown and back, right? Then all the roads going from the suburbs to downtown would all be a big tree, right? Like, but actually you have fluctuations in the demand, right? Like, there's some demand to go downtown, but there's also occasionally demand to go to your neighbor, or to go to, your family, one town over, or to go to the grocery store, or to go. So you have fluctuating demand, which means that, sometimes people want to go, resources and sinks are all kind of moving around, and so then you get, road networks that have loops in them, thankfully. Okay. Okay, so now back to the 3D, right? We want to think about 3D. The example I showed before was in 2D. And we also want to go back to the sort of ensnarled network idea where we have, two different networks sharing space, right? And so another way that the spatial character can come in is if, and again, now this is inspired by the liver, so you have the green as the bile, the purple is the blood. And for this case, we're going to take a super simplified, highly symmetric mesh, just a cubic mesh for both networks. Stick one and install into the other, and then say, okay, well now we have, for every edge, every pipe in the system, right, cuts, directly through one loop to the other network, right? And so one can think about how close together the pipe wall is of one network to the other, and then include a distance dependent, a repulsion or an attraction based on the proximity of our state. And so in the liver, right, the space in between the networks is not open space, it's mostly 90+% hepatocyte cells. And the blood network corresponds to the apical surface, so one of these sort of special surfaces of the hepatocyte cell that has sort of a different intracellular machinery. And likewise, the bile, I'm sorry, the other way around. The bile is the apical surface and the blood is the basal surface. And so these two sort of specialized surfaces in a polarized cell that have different biological machinery. And it has been shown in much simpler cases than this, that the sort of family of paraproteins can generate, attraction or repulsion between the apical and basal surfaces of a cell. So there is a biological mechanism that exists that could generate an attraction or a repulsion in exactly this way. So it's not completely crazy. Also, you might want a biological system might want to have some kind of spatial regulation of where these are, because of the fact that you want to repel when you're close because you don't want bile to possibly accidentally get into your blood, but you don't want to get too far away because you are performing a filtering job, right? Like one is filtering into the other, so you want to maintain some sort of proximity. So anyway, you can write in a distance dependent coupling, right? Basically just think, it's a sort of lower dimensional because it's all tubes, but it's basically like a gravitational or electrostatic coupling, essentially. It's really nothing more complicated than that. And then include that in the update term, right? So now we have the shear strength, the overall cost, like the upkeep cost of the network, and now the coupling interaction. And we're going to look at, I said before, you could sort of look at how many loops are left versus how many you have. That's what this nullity is, right? So the nullity is how many loops do you have at time infinity versus how many you started with, how many cycles. Okay, and so we can look at these sort of phase plots. The one on the left is for a negative coupling, so repulsion between the tubes. The one on the right is for attraction between the tubes. And the x-axis of both of these is the fluctuation strength. The y-axis is the strength of the sort of spatial coupling, the repulsive or attractive coupling. And then the color scale is how many cycles did you stabilize, right? And just to be for clarity, I should point out, and this is maybe slightly bad form because they're next to each other, but this yellow is a lower value than this one. So this yellow really goes almost 100%. This one only goes a little under 50%. With some example, 3D networks shown. And so the interesting thing here, right, is that at low values of the coupling, so essentially you go back to the case where you just have fluctuations, right? You see here's the fluctuation phase transition, the same as before. And you can start turning on the coupling and like nothing happens and then you start bending over where the location of the phase transition is, right? You make it, harder and harder for the fluctuations to stabilize, loops in the negative coupling case. And interestingly, in the positive coupling case, you don't really do anything to the normal fluctuation topology transition, but you create a whole new transition that sort of washes it out for a significant, a certain value of the coupling. All right, and so, once the coupling gets high enough, suddenly the coupling just completely dominates everything and you stabilize all the cycles. You want cycles everywhere. Whereas the fluctuation is only capable of stabilizing, at most half of the possible cycles. Okay, so the fact that, you now have, spatial terms and spatial ideas connected to the topological states, has strength, right? There's real power here for, the molding of a topological state, right, in these complex networks by biology. Now, whether or not biology does exactly this, we don't know yet, but at least the potential is there. I would just go over this really quickly, but like the form of the transition, you have this really, for the repulsive coupling, you have this really nice curve collapse. So it really does look like, a phase transition. For the attractive coupling, it's a little messier. There's more kind of interesting detail in where these plateaus are, but there's a broad sense of a curve collapse anyway. Okay, so now let's talk about some actual data. So that was all just theory. So this, hopefully you can see this on the screen. This is a visualization of the liver tissue. Purple is the sinusoidal blood networks. The green is bile, that you're seeing now. This is biocaliculi. These are the nuclei. These are the nuclei of the cells, the hepatocytes that are in between. These are the surfaces of the hepatocytes. These are the volumes of the hepatocytes filled in. This is the two networks together at the same time. Very, very rich structure, and the nuclei included, surfaces included, and I think some bodies included, yeah. Okay, so like lots of different layers of data in 3D. We're going to concentrate on the networks. This is from courtesy of the Zarial Lab and the MPI-CBG, and also now the Human Technical Milan. So that's the liver. We're also going to talk a little bit about the pancreas. So the pancreas is the adult pancreas is quite different than the liver. So the liver has these two, completely ensnarled systems. The pancreas, at adult times, is more, at least the ductal network of the pancreas, where the exocrine endocrine is secreted. It becomes a tree network, right? And so it's a little bit different. But early times, it's not like that. And you have many different cell types arranged around the duct. So you have these acini, exocrine and endocrine acini that do the secretion. You also have, different, cells for lighting the different ductal regions. And so just to give an idea of what the data is like, the different stages, right? So this is work done by Colleen and Young Ho and on Tom's lab in Dresden. They do this 2-photon imaging, and then they segment. So network segmentation in 3D is a nasty job. But they do the segmentation, they do skeletonization, and they convert it to a graph, right? So there's these multiple steps to ultimately get a network out of it. And then they look at, and in this particular case, they're parsing out the cyclic part and the tree-like part. That's the darker blue and the lighter blue. The dark blue is the cyclic part, the lighter blue is the tree-like part. Okay, and they, like I just said, at adult times you get a tree, and this is work that's pretty old at this point from the lab, actually before they were in Dresden, before I knew any of them. They showed, the sort of time course and development of the pancreas in the mouse that goes from sort of hyperconnected to tree-like, and they also have some sort of organoid, complementary organoid sort of data that sort of always, is very, very sort of hyperconnected and complex. Organoids have no, coherent flow, right? They have no outlet, whereas the actual organ has, an outlet through the duodenum. Okay, and so we can look at the network structure and try to understand some things about pancreas development, right? In the light of sort of thinking about these sort of flows and the resistances and conductances and everything else. So one of the obvious things is the whole structure is growing. Right, so the total network length, the edge count, everything is going up, going up quite a lot. So I should point out, by the way, that like in early times, the pancreas has those dorsal and ventral parts that they sort of image and analyze separately. So the left, the bigger thing on the left is the dorsal part, and the smaller part on the right is the ventral part. One can also lump everything together and look at it all together as well. But they don't do that because, for each of these data points, they have to, extract, kill the animal, extract the data, but it's not, they can't follow the same organ, right? They have to take snapshots. And they can't do, it's difficult, I think, to do the same side. I think they do the same side sometimes, but it's, especially the larger ones is tricky. So they separate them. Okay, and so then if you look now at the fraction of the network that is cyclic, right? So you have a whole bunch of network, and you look at the fraction, either by edge or by length. Here it's by length. You can also look by edge, it does the same thing. You have, even from early times, the fraction is maybe growing, but relatively stable for quite a while in development. And then that cyclic fraction suddenly, over the last 2 days, essentially, completely collapses, right? So it's known that you are definitely having new networks, new, sprouting of branching points all throughout this time, right? And so that means that in order for the cyclic fraction to be stable, if you're growing the network and making sprouting new branching points everywhere, then you must also be creating new cycles, right? You have to be filling in more and more cycles. And if you look at the cycles over time, that definitely goes up. So you're creating more cycles, but at a sort of a constant ratio, essentially. And so there's somehow both creation of cycles and pruning of cycles going on in balance, and then the creation goes away and the pruning dominates somehow. But if you look at the edge lengths, right, and you think about what's going on, and the edge length is a sort of marker of thinking about the overall flow plot profile of the network and thinking about how one would build a network like this. You look at it looks like it has this, there's no interesting structure. It looks like an exponential, just by eye. One can actually quantify that, you plot it in log space, and it's a perfect straight line. It is an exponential. It's very, very, very surprisingly close fit to an exponential distribution at almost every length scale. Like the very, very, very tiniest ones, you fall off a little bit from what you expect. The amount of data at the really, really long edge lengths is really low, so this is noise. But it's it's the same story at sort of more or less all times. Even after the system starts falling apart, you get the same thing, but the slope changes. The slope is just the mean edge length, but it stays exponential. So other networks don't have this property, right? So the pancreas is sort of special here. If you look, for example, at the slime mold I mentioned before, or the liver that we've talked about a few times already, and you only look at the cyclic part in the same way, then the tail in both cases is pretty exponential, but you have to go already to very high edge lengths before you reach that tail, and before you reach that tail, it's very, very far from exponential. It's doing something else completely. Okay. So one thought, right, this is a bit suggestive, we're still writing this up at the moment, but an exponential distribution is what you would get from just pure random subdivision of edges. So that if you have something sort of like the equivalent of a Poisson process, but not for points, but for partitions of a network space or growth of a network space, together with then just an overall inflation, would also is one way to generate an exponential distribution. And so that would indicate that there's no constraints. Like it's just subdividing edges. It's growing one tip into another tip, willy-nilly, without actually any specific guidance during this stage, and that then adaptation would kick in later, right? And sort of normalize everything out into something that's sort of more structured. And so that's the prediction that would come from both looking at the data and knowing how the adaptation steps work. And so that's where we are. This is work in progress with the pancreas. With the liver, I already showed you these things. I already showed you this. I just want to, because it's past the time for break, I just want to jump quickly to, I already showed you this. I just want to get this comparison, right? So there's this kind of interesting, you have these two different organs, that both have really interesting, pairs of networks and relative pairs of network structure, right? So both organs have a cycle-rich blood network at all times. Well, I mean, at early enough and late enough in development, and all, but beyond that at all times. The liver has a cycle-rich embryonic bile network, and it stays cycle-rich into maturity. Whereas in the pancreas, you have a cycle-rich embryonic ductal network that falls apart into a tree at maturity. You get different edge-length distributions. Remember, yesterday I showed you that the edge-length distributions in the liver have this hidden bimodal character. If you look at the beta skeletons, if you look at the beta skeletons in the pancreas, it just stays; those also just stay exponential. So in beta, you have this hidden bimodal edge-length distribution that does not appear in the pancreas. The actual edge-length distributions of the network itself, if you don't look at beta, stay perfectly exponential in the pancreas and have this strong departure from exponentiality in the meat of the distribution for the liver. Excuse me. Both networks have this property of being ensnarled with each other early, but only one of them stays ensnarled, largely because the other one falls right on the tree. And so, yeah, there's some interesting similarities and differences. One thought that we have is that, the early time developmental program for both networks, for both systems could be the same, and then something changes in the liver. You remember the liver has this non-monotonic profile in the ensnarlment in time, but something changes in the liver to stabilize the cycles in the second network that doesn't happen in the pancreas. And so again, I would advertise the postdoctoral for this program. And then let's have a break. I should point out, by the way, there's also a faculty visitors program associated with it. So if you have a sabbatical or something coming up, or want to come to Dresden for a few weeks, then we have support for that too. So not just for potential postdocs, but also for faculty that you may be interested in coming to Dresden for a while. Check it out. Okay, let's have a quick break.
Yeah, 15 minutes.
Yeah.